nice thing is this is a PC just like mine. You don't have to do the Apple PC trick. As you said, my name is Tim Fouts. I'm with a small company named Perfectus Bioscience, and then we'll talk about some work we've been doing on a particular HIV vaccine that is entering into clinical development. And the subject of the talk is how the story behind that particular vaccine candidate, and some observations we've made. And the title is the title of a recent publication we put out this past April, I think, in PNAS. So for those of you who don't know who Perfectus is, we're a small clinical stage biotechnology company, and we work on a variety of viral diseases. Some of the big ones including Ebola Marburg, as well as HIV. And we have efforts in chikungunya, HSV, and other biodefense targets, including alpha and arenaviruses. Oops. Okay. So principal technologies involved at Perfectus include a virus, a recombinant virus, VSV, vesicular stomatitis virus, DNA and adjuvants, as well as subunit-based technologies, including the one I'm going to talk about today, which we call transition state-based vaccines. Okay, so the candidate I want to talk about today is one we call full-length single-chain, FLSC. And what it means, it is a fusion protein between HIV envelope GP120 and its principal receptor, CD4. And there's a linker between the two that allows the combination, the GP120, to refold itself to mimic the transition state that occurs as the HIV enters the cell. So starting out with binding to CD4, undergoing a conformational change that allows it to interact with its co-receptor. And is this form, this shape, that we're trying to mimic with this construct, this transition state construct. Okay, so the reason we did this is we knew that from a lot of work that chronic viruses tend to hide their vulnerable parts, and HIV is no different. So we felt that if we altered the envelope structure using chimeras of the envelope and its key receptor, would change how the antigen is presented to the immune system and enhance those parts of the conserved vulnerable areas, and enhance their immunogenicity and potentially expand the epitope diversity in the antibody responses that could be protective in vivo. Now, there are a number of structural analysis and molecular models that I think have been published in Science and Nature regarding the shape of the HIV envelope and how it enters the cell. And so many of these models have suggested that these CD4-induced, CD4-induced epitopes may not be as easy to get to by an antibody. But there are a number of papers out now that suggest that perhaps that may be, in the real world, that may not be correct. So antibodies to transition state epitopes, mediate potent ADCC activity against cell-bound virions. This is published by Guan and Lewis in 2013, as well as others. In a number of non-human primate studies link protection from infection with antibody specificities, including those that directed to CD4-induced epitopes and functions that lie outside the sphere of direct virion neutralization. So we're talking about protection is mediated not by neutralizing the virus directly, but perhaps by killing the infected cells. How might this be possible? Well, we know that aside from the native, natural envelope structure, there are a number of other structures of envelope that appear on infected cells and on the virus itself. And many of these are not really viable in terms of mediating viral entry. However, they may be very adequate in terms of targets for ADCC or other Fc-mediated antiviral activities mediated by antibodies. Okay, so we undergo a number of studies to try and prove that full-length single chain could potentially protect against infection. And one of our first papers we did this, was published in PNAS a number of years ago. And basically it used 4 different groups, one in which had the rhesus form of full-length single chain, RHFLSC, in which case we swapped the human CD4 for rhesus CD4, and all maintain the same shape antigenically as the full-length single chain. Now, you don't have to worry about the other groups so much, as there were other test candidates we were looking at as control, including BAL GP120 and human CD4. All were mixed in QS21, which at the time was the adjuvant du jour. Immunized the animals several times, and then began a challenge with a single high dose of SHIV 162P3. For those who may not be familiar with these models, SHIV is a chimera between SIV and HIV, such that the envelope protein of this virus is HIV, while the rest of it is SIV. Okay, now, in this high-dose challenge model, what we're looking for is that the vaccinees have a lower viral peak and set point as compared to controls. That's our key measure of success. And you can look at this as a sort of an area under the curve measure and to compare the different groups.
I just need to interrupt you for a sec. We're having a technical intro. I just need to get a tech guy up here for 1 second.
Oh, sure. Pause for technical interruption. Let me know when you want to start. When you start. Oh, good? Okay. Okay, here we go. All right, so again, all right, whoops. This thing is a lot more sensitive than I expected. Okay, anyway. All right, so through this study, we identified a relationship between lower viral set point, again, that was our key measure of success, and higher titers to CD4 antibodies. And so in this case, in here, group that immunized with rhesus full-length single chain compared to all other groups had lower viral set points is measured by viral load area under the curve versus all the other groups. And that group also had a much higher CD4I antibody titer. And this particular antibody we're using, N12I2, was identified by George Lewis at the University of Maryland. And it's a very typical CD4I antibody. Okay, so that was promising. All right, now, so of course, as the time progressed, since that paper was published, the model systems that people use to evaluate vaccines against HIV changed from one where you do a high-dose viral challenge to now where you do multiple low-dose. And the principle here is that since HIV is transmitted at very low doses, and so we thought the principle is that if you do multiple low doses of infecting virus such as SHIV or SIV, that it more closely mimics how HIV is actually transmitted. So in this case, protection is offered by the number of challenges it takes before the control animals become infected compared to vaccinees. And so you hope to see some distance on a Kaplan-Meier curve between when the animals begin to get infected as an indication of protection. Okay, so our initial experiment we tried was very ambitious, and we was powered with 6 animals per group to detect greater than 82% protection. In other words, probably overly ambitious slam dunk. But the principle here, we used a single rhesus single chain mixed with 2 different adjuvants, RC529, which is a squalene glycerol mix with monophospholipid A, and ISCO matrix, which was a saponin-derived ISCOM, compared to GP120 as a control. In this case, the animals immunized several times, and then after 4 weeks, they were challenged weekly with a fairly low dose of SHIV 162P3, one of the prevailing SHIVs that at the time. 50 TCID 50 is pretty low, and generally you don't expect the animals to become infected, the control animals to become infected after 3 or 4 challenges. Okay. All right, so this is the result of all the challenges with all the groups here. And again, this is published, but it's really not, this was a little discouraging at first. None of the groups seemed to be any better than the others. Maybe single chain RC529 group seemed to be a bit better. It came close to significance, but not quite. So there's a trend, but not clearly not a slam dunk. Now, we did notice that animals that were immunized with rhesus full-length single chain all did have a fairly good response to CD4-induced epitopes. And these include these antibodies down here, up here, 17B, A32, 19E, M9. These are all antibodies, and they all recognize different CD4-induced epitopes on the envelope. So, and as you can see, there's a very nice response with the animals immunized with single chain against these epitopes, not so much with GP120. In contrast, with the soluble CD4, very little response against this epitope from rhesus folding single chain animals, while the GP120 animals had a very nice response that competed with this binding of soluble CD4. So again, altering the shape of the antigen changed how the immune response performed. Okay, well, so we asked the question, well, what if you were to dichotomize the animals that were immunized by their titer to the CD4I antibody? In this case, we selected N12I2, you saw that before. Well, there was above or below the median, and what we found was that the animals that had a higher competitive titer against this epitope were better protected versus those that had a lower titer. In contrast, animals which presented titers to CD4 binding domain titers greater than median or less than really were no difference. So in this, there was clear benefit for having antibodies against the CD4-induced epitope. Now, we also know that HIV is a interesting virus in that it knocks out the cells that are the principal drivers of the immune response, and those being the CD4 positive T cells, and those in particular those express CCR5. So we asked the question, well, was there a benefit perhaps for having too many or too little T cells in this response? So what we did is we looked at IL-2 spot-forming cells, in a LE spot, just simple LE spot, and those that expressed IL-2 above and below the mean. And what we found was that the animals which had a lower IL-2 LE spot response had a much better ability to control, prevent infection, rather than those which had a very high IL-2 LE spot response. Now, interestingly enough, this one long bar here is a single animal that was immunized with folding single chain. Okay, so perhaps protection was apparent, but was lost because there were simply just too many T cells, too many targets. So we then asked the question, well, what if you were to remove the animals that had that high T cell response and then reevaluate, were there any protection offered by any other immune measure in the animals which had the low T cell response? And lo and behold, animals which we saw a very nice correlation with folding single chain titer, ADCC titer, and competitive titer to another CD4I antibody, A32. And this one was particularly interesting because this was a key correlate measure in the RV144 trials. Okay. Or one that was kind of interesting. Okay, so this suggested to us that there was a sort of a zone of immune balance, and this is just a diagram to express what our thoughts are here, where you have LE spots going one way, ADCC going the other, and the color depicts the number of challenges it takes to get infected. So you could envision this sort of zone where there's a good chance you'll get protection if you have the right balance of ADCC response and not too many T cells. Okay. Now, we did another study, again, trying to validate what we had observed in our initial study, this time using single chain versus another group that had single chain mixed with TAT toxoid, which we thought would be an interesting antigen. It was a subunit-based antigen. It had some evidence that it was offering some protection in certain studies. Mixed with another adjuvant, and again, this is just because this was what was available to us, this saponin-derived adjuvant called GPIO100, mixed with a TLR agonist. Okay, so in this case, the study was a little different in that we used a SHIV challenge dose, and we had to increase the challenge doses as we went along because the animals just weren't getting infected. So we had to increase until a final dose of 200 TCID50s. So the key differences including TAT was one of the adjuvant One of the immunogen formulations, we used a higher concentration amount of single-chain vaccine. That was just because we knew from our preliminary work that the adjuvant wasn't as good as some of the other adjuvants we had worked with in the past. So we had to increase the dose, single-chain, different adjuvant, and increase the challenge dose over time. All right, so here's our Kaplan-Meier curve. You can see animals that had the single chain alone seemed to do better, while the animals that had single chain and TAT toxoid didn't do as well. And interesting enough, this animals that had single chain alone had a higher ADCC titer, including total titer and plateau cytotoxicity. TAT toxoid, interestingly enough, increased T cell responses, and later we found out that TAT toxoid actually associated with the single chain, and probably, for those of you who don't know anything about TAT's really good about transcytosing proteins across cell membranes, and so we speculated that perhaps what this did, the toxoid wasn't completely devoid of that activity and was able to move the single chain across the membrane and able to access these compartments that were able to present to T cells. So you can imagine this an immune balance in the protected group where you have ADCC over interferon gamma responses much higher than the animals that had TAT toxoid. Okay, so that was 2 studies using SHIV 162P3. We switched models again, just again to prove that we weren't fooling ourselves. In this case, we used a different model. In this case, SIV MAC 251, very robust challenge. The study was powered to detect 75% efficacy with 8 animals per group. In this case, we did something a little different. We did a DNA prime subunit boost regimen because we wanted to see if we could actually promote ADCC responses. In this case, we had a single chain and a adjuvant and a plasmid expressed GAG-PAL, either without adjuvant, a genetic adjuvant derived from heat labile toxin A1, rhesus IL-12, and a combination of the two. The reason we picked these, so key differences, we used the MAC 251. In this case, this was one that had a very high quasi-species diversity, challenge virus antigens derived from SIV MAC E543. Now, in the parlay of SIV, this is the reason we picked this particular strain. This is compared to the challenge strain, it is represented, the difference between the two represents a cross-clade challenge. So it's a very robust challenge. DNA prime protein boost, and adding IL-2 and LTA1 as genetic adjuvants. Okay, so real quickly, IL-12 is interesting. Perfectus has this adjuvant, genetic adjuvant, in clinical trials. It's performed quite nicely. It induces, we were using it initially to induce T cell responses, but we also know it drives ADCC. It's safe and well tolerated and active in clinical studies. The LT-CTA1, interesting because it causes immune activation because of amplification of cyclic AMP. And we knew, well, this is our construct where we delete to those who know something about these A subunits of these heat labile toxins, know that it's a 2 subunit toxins, so we deleted the B subunit as well as 2 pieces of the, 1 piece of the A subunit to just have this A1 piece expressed as in a plasmid. And we knew combinations of IL-12 and LT1 increased the LE spots. So our speculation was, well, although it increases, the combination increases LE spots, could it also increase ADCC? All right, and this was published recently in Human Vaccines and Immunotherapeutics. Okay, so at least our observations with LTA1. Okay, so here are the Kaplan-Meier curves, and we noticed that inclusion of IL-12 in the prime step yielded about 76% efficacy in the whole study. So here are the red, you can see IL-12 alone versus DNA alone, IL-12 versus all the other groups. And then we had a number of collaborators who had animals that they used as controls where they received the same SIV MAC 251 challenge virus, the same dose, administered at the same time by the same technical staff, or within the same 1 year of the start of our challenge protocol. So we felt it was valid to include these as controls, and this is where we get the 76% efficacy. Okay, so we noticed that we looked at LE spot responses, that indeed the group that had the highest LE spot response was the one that had IL-12 and LTA1. IL-12 also increased ADCC, which is what we were hoping for, both in the group with IL-12 alone and the combination. All right. Now, so the question now is, okay, how does that parse out in terms of protection? What is the relationship between ADCC and the T cell response? So in this case, the way we looked at this is we decided to partition the animals into those that had no evidence of ADCC versus those that were positive for ADCC, and divided the interferon gamma LE spot response into quartiles. We noticed that the animals that had in the first 3 quartiles, in less than 75th percentile, were much better protected than those animals which were either ADCC negative, the blue, or were positive for ADCC, but it had a very high gamma interferon response, and that's the green. Again, this idea of immune balance. So yes, if you, does single chain protect? Yes, if you induce a CD4I response above a certain threshold, you have evidence of ADCC, and without an overabundance of T cells. And it's this concept of immune balance. And this was what we published in a recent PNAS paper. So our goal in our clinical studies is to try and improve the potency and durability of this FC-mediated protection with the right balance of supportive T cells. Because we know we need T cells to get the antibodies, but we just certainly don't want too many. Okay, so as I said, this single chain is entered into clinical trials. We have a released product. Clinical trials have just started. Drug substance is GMP manufactured. We're getting about 1 gram per liter final. Drug stability at 20 minus 20 at 12 months. Labeled product release. We've completed all the standard tox studies, and we also had to perform a non-human primate immunotox study. As you imagine, since the product has a piece of CD4 in it, the question was, are we inducing any deleterious auto-CD4 responses? We did the study to try and prove that we were not, and there was not any evidence of any kind of deleterious auto-CD4 responses induced by single chain or its rhesus partner in the animal. Okay, this is all good. Currently, we have about 3,400 vials of the drug product and 125 grams of the unformulated material available for future studies in collaborations with the interested parties. You can always talk to us about that. We're always interested in working with others. Okay. Phase 1 clinical trial, as I said, is ongoing. 3-dose study starting at 75 micrograms, ending up at 300 micrograms. Individuals will receive 4 doses of full-length single chain. Looking for, obviously, a primary endpoint of safety and tolerability. Secondary endpoint would be antibodies to full-length single chain in about 80% of the vaccinees, and competitive serum titers to CD4I antibodies in approximately 60% of the vaccinees. And we'll be looking at fluctuations in CD4 positive T cell counts over time as an indication of whether we're inducing any kind of deleterious response to CD4. Again, exploratory clinical trials is an older slide. You can imagine we'll need to look at a number of different options. Viral prime, the VSV is just an example. DNA prime, other adjuvants potentially. So this is hopefully something we'll eventually get to once we get past. So here are the acknowledgement slides of all the individuals that have contributed to this effort, including those colleagues at the Institute of Human Virology who have been instrumental at doing this, including my mentor, Tony DeVico, and George Lewis, Bruce Gillum, who's currently running the clinical trial, our leader, fearless leader, Bob Gallo, members of my team at Perfectus Bioscience, Jennifer Swartz, Ken Bagley, Leah Prado, and Kate Bob. And then funding, of course, would not be possible. Bill and Melinda Gates Foundation award to the Institute of Human Virology, SBI awards and R1s from the NIAID and money from the Henry Jackson Foundation. So thank you very much.